Welcome back to our YouTube channel. So, without wasting time, let's start our topic culture of viruses. Number 1, viruses are extremely small infectious agents that invade cells of all types. So, viruses are basically very tiny particles that can enter in cells of all types. Number 2, viruses are obligate intracellular parasites, so they depend on host for their survival. They will always need a host for their survival and multiplication. Number 3, viruses cannot be grown in non-living culture media or on agar plates alone. They must require living cells to support their replication. Viruses can't grow in non-living culture media. They will need a host for their multiplication. Number 4. Viruses have been the culprits in many human diseases including smallpox, flu, AIDS, etc. Techniques in cultivating and identifying animal viruses. Viruses require living cells as their medium. In vivo, we use lab-bred animals and embryonic bird tissues, and in vitro, we use cell or tissue culture method. The primary purpose of virus cultures are, let's see, number 1, to isolate and identify viruses in clinical sample. Number 2, to do research on viral structure, replication, genetics, and effects on host cells. So number 2 point says that we need the virus cultivation so that we can study the structure of their cell wall and their DNA structure and how they affect on the host cell. What impact does they create on the host cells? Number 3, to prepare virus for vaccine production. So this is most important. We need them to culture so that we can prepare the vaccine. Nowadays we take COVID vaccine to prevent us from the deadly coronavirus. So it is the one of the purpose for virus cultivation. Methods of cultivation of virus. Generally, 3 methods are employed for virus cultivation. Number 1, inoculation of virus into animals. Number 2, inoculation of virus into embryonated eggs. Number 3, tissue culture. So let us discuss one by one them in details. Firstly, animal inoculation. Viruses which are not cultivated in embryonated eggs and tissue culture are cultivated in laboratory animals such as mice, guinea, pig, hamsters and rabbits are used. So viruses which we do not cultivate in the embryonated eggs or in tissue cultures, we use which method? Animal inoculation. And we take the lab-bred animals like mice, guinea, pig, hamsters, rabbit, etc. Number 2, selected animals should be healthy and free from any communicable disease. So this animal should be free from any spreading diseases. Number 3, mice are most commonly used. Number 4, after inoculation, viruses multiply in host and develops diseases. The animals are observed for symptoms and disease and death. After the inoculation, the virus get multiply in the host organism and we can see and we can observe the symptoms and disease of the animals. Number 5, then the viruses is isolated and purified from the tissue of these animals. So after observing those symptoms, we isolate them from the organisms to study further the viruses. The advantages of this animal inoculation method is, number 1, diagnosis, pathogenesis and clinical symptoms are determined. Number 2, production of antibodies can be identified. Number 3, primary isolation of certain viruses. Number 4, mice provide a reliable model for studying viral replication. Number 5, used for the study of immune response, epidemiology and oncogenesis. These are the advantages. Now we come to the disadvantages. The disadvantages are expensive and difficult in maintenance of animals. Number 2, difficulty in choosing of animals for particular virus. So sometimes it becomes difficult to choose which animal for which virus. So it is one of the disadvantage. Number 3, some human viruses cannot be grown in animals or can be grown but do not cause diseases. So some viruses do not grow in animal cells, and even if they grow, they do not show any symptoms of the disease. They do not cause any disease. So it is one of the disadvantage. Number 4, issues related to animal welfare system. Now the second method, inoculation into embryonated egg. Good Pasteur first used embryonated hen's egg for cultivation of viruses. This is the name of the scientist Good Pasteur, who used first the hen's egg for the cultivation of virus. Process of cultivation of virus in embryonated eggs depends on the type of eggs which is used. Number 3, viruses are inoculated in chick embryo or 7 to 12 days old. Number 4, for inoculation, eggs are first prepared for cultivation. Shell surface is first disinfected with iodine and penetrated with a small sterile lid. So, for inoculation, we have to penetrate the egg with a small sterile drill. And number 5 is, after inoculation, the opening is sealed with gelatin or paraffin and incubated at 36 degrees Celsius for 2 to 3 days. The inoculated egg is then sealed with gelatin or paraffin so that some foreign particles do not enter in the egg, and that egg is incubated for 36 degrees Celsius for 2 to 3 days. Number 6, after incubation, egg is broken and virus is isolated from tissues of egg. Number 7, viral growth and multiplication in egg embryo is indicated by the death of embryo, by embryo cell damage, or by formation of typical pokes or lesions on egg membranes. So how we can determine that the viral growth and multiplication has taken place? By looking at the cell of the embryo. So we can see the death of the embryo, or we can see the embryo cell damage, or we can see some typical lesions on the egg membranes. So by this we can find out that the viral multiplication has taken place. Number 8, viruses can be cultivated in various parts of eggs like chorioallantoic membrane, allantoic cavity, amniotic sac and yolk sac. So these are the various sites where we can inoculate the virus. I have drawn a diagram here. So this is chorioallantoic site, and here we can inoculate, and it is called as chorioallantoic membrane inoculation, and this is amniotic inoculation, and here is the yolk sac, so yolk sac inoculation, and here is the allantoic inoculation. This is the diagram of embryonic egg which is inoculated by the viruses. Chorioallantoic membrane. Here inoculation is mainly for growing pox virus. And number 2, herpes simplex virus is also grown. This method is suitable for plague studies. Allantoic cavity. Inoculation is mainly done for the production of vaccine of influenza virus, yellow fever and rabies. Most of the avian viruses can be isolated. Amniotic cavity, mainly done for primary isolation of influenza virus and mumps virus. Yolk sac. simplest method for growth and cultivation of virus. Most of the avian virus can be isolated and detected. Advantages of this method are: number 1, widely used method for the isolation of virus and growth. Number 2, ideal substrate for viral growth and replication. Number 3, isolation and cultivation of many avian and few mammalian viruses. Number 4, less labor is needed. Number 5, widely used method to grow viruses for vaccine. So these are the advantages of the egg inoculation. Now we see the disadvantages of this method. The site of inoculation varies for different viruses. So for different virus, we use different parts of the embryonated egg, like we use sometimes allantoic site, we use sometimes yolk sac, we use sometimes amniotic cavity. So these are the various site of inoculation of viruses, of different viruses. That's each virus have different site for their growth and replication. Number third method is the tissue culture method. In here, cultivation of tissue or organ for growth of viruses is done. So basically there are 3 types of tissue culture method. Number A is organ culture, number B is explant culture, number C is cell culture. So in organ culture, it is mainly done for highly specialized parasites of certain organs. Example, tracheal ring culture is done for isolation of coronavirus. So for this coronavirus, we use organ culture method to cultivate it. So in explant culture, we use what? Fragments of tissue can be grown as explant. Number C, cell culture. It is mostly used for the cultivation of virus. So here it is mostly done for the cultivation of virus, and this is the cell culture. It is mostly used. Okay. Now the advantages are: relative is broad spectrum, cheaper and sensitive. Disadvantages of the tissue culture method are: the process required trained technician with experience in working on full-time basis. So we need a lab technician to operate this tissue culture method. Number 2, tissue or serum for analysis is sent to central laboratories to identify viruses. So here we end today's video. Hope you understand the topic tissue culture. Thank you.